proteins have two or more separate polypeptide chains linked non-covalently, each of which fold into a complete tertiary structure. That is called a subunit. The spatial arrangement of subunits and the nature of their interactions constitutes quaternary structure. The individual subunits have no biological functions, only those held together to form quaternary structure have. The subunits of a protein may be identical or different. If a protein has two identical subunits, it's called a homodimer. A protein has two different subunits is called a heterodimer. A multi-subunit protein is called a multimer. A multimer with just a few subunits is often called an oligomer. A multimer consisting of one or more identical subunits is referred to as a protimer. For example, human hemoglobin, the oxygen-carrying protein in blood, consists of two alpha subunits and two beta subunits, all four held together by non-covalent interactions. Each alpha subunit is paired in an identical way with a beta subunit within the structure of this multi-subunit protein, so that hemoglobin can be considered either a tetramer of four polypeptide subunits or a dimer of alpha-beta protimer. Many proteins have multi-polypeptide subunits. The association of subunits can serve a variety of functions. Many multi-subunit proteins have regulatory rules. The binding of small molecules may affect the interaction between subunits, causing large changes in the protein's activity in response to small changes in the concentration of substrate or regulatory molecules. The exception is that a few proteins contain two or more polypeptide chains linked covalently. For example, the two polypeptide chains of insulin are linked by disulfide bonds. In such cases, the individual polypeptides are not considered subunits, but are commonly referred to simply as chains.